All right, good afternoon. I'm sorry if I fooled anybody yesterday, making you think that you were done. Sorry, you're not. I wish we were all done. Don't get me. I mean, I enjoy being here, but I also enjoy, being in my office with a cup of coffee. So, okay, so we have two more. Thank you. We have two more sections or two more topics. One being recombinant DNA technology and the other one being sequencing. These are things that we do in the lab all the time. So the technologies that I'll work through with you guys is something we use in the lab all the time. It's, some things we've modified as new enzymes become available, but these are the basic techniques that we use to do the work that we try to do more or less successfully. So we'll work here, your objectives, blah, blah. We'll work through these different sections here, these different techniques, and we'll talk about all these different things, but not everything is going to be covered today. So today's topic 22, will probably be more 2 lectures, and then we have a final one on sequencing next week. Okay, so we will start, funny enough, we'll start with RNA and not with DNA, because the example, I'll use an example to walk you through all these different techniques. And the example that I'll be using starts with isolating RNA from humans in order to find the cause of a specific disease. So this is your disease, and really it's just an example. You could use any other enzyme that you want to work with or any other protein that you want to work with, and you would get a similar mode of what you're going to do about it. So the disease is a family with language impairment. Some of the family members in black here, the filled circles and squares, have symptoms of simplified speech, poor awareness of appropriate sound pattern, poor articulation, and they struggle with complex sentences of tongue and movements. And when you see something that occurs in multiple family members, it's likely that it's something genetic. So it's not it could be a learned behavior, but if it's in one sibling but not another sibling, it's more likely that it's a genetic factor that plays into this. This is your pedigree. I don't know if you've learned this stuff in genetics, but basically you have the parents. The parents had 5 children here. Of the 5 children, 4 had the language impairment, 1 did not. And then of the ones that did have language impairment, some of their offspring, I don't know how they had that many kids, but some of their offspring, I have 5, so that's hard enough. I don't know how you deal with this kind of stuff. Like, I have no idea. Anyway, so some of them had the disease and some not. So the way you go about this is what you want to do is you want to try to figure out what's wrong by sequencing the DNA and then looking what the error does. Okay, so if you have a mutation in the DNA, as you remember, any mutation can be a silent mutation, a missense mutation, and even it can be a frameshift mutation. We had all the different kinds of mutations, but you don't really, if you just see it in the sequence, even if you see that it's missense mutations, you still don't know if that has any effect on the protein itself. If it's somewhere on the outside where it doesn't interact with anything, maybe that mutation makes no difference at all. Or maybe it's a super important amino acid that gets mutated. But you'll have to go through all the biochemical processes and the genetic processes in order to figure out what's actually going on. So it is, in our case, already known that there is a gene called FOXP2 that's related to speech and language disorder. We also know that it's located on chromosome 7. And we now want to know, is this FOXP2 gene involved in this specific language disorder? We don't have, when you start out, you have the family, but you haven't looked at their genome yet. So you don't have the information whether there's mutations in their genes or not. So what you want to do is you want to get the DNA sequence of that gene. You also may want to, if you find a mutation, may want to purify the protein and investigate its properties. And in the end, you may even want to generate transgenic organisms. So if I change this protein in mice, does that make a difference? Is it important? Mice don't really speak, but they have other other patterns that you can observe. You could, mutate it in any other organism that's a good model organism for the disease you're looking at. So what we want to do is we want to clone the gene, and the clone is a copy of the original. So we want to take the genetic information from the human and put it into a format that we can work with. In the lab, we often work with Escherichia coli, and I've mentioned this one before. It's our little pet bacterium. It's a very fast-growing enterobacterium that's super easy to grow in the lab. So all you need is some media. So media is in this case has yeast extract and all kinds of stuff that makes the bacteria happy. It has everything they need: amino acids, nutrients, vitamins, whatever E. coli needs is in that media. And then you grow it at 37, and it is it's super easy to work with, and it grows beautifully every time. So we use E. coli for cloning, and there's many different tools available for E. coli that we can use in the lab. Okay, so I'll walk you through all these different sections on how to get from trying to get the sequence information, synthesizing the DNA, making an expression construct, to in the end purifying the protein from E. coli. That will be our workflow in the lab. And this is a typical workflow that I would give a master student. Okay, so somebody who's wanting to work in the lab for maybe a year or maybe 2, that's a long enough time to get all these things done and get a good characterization out of it. It's not something I would give somebody who works in the lab for 4 weeks. So if you work in the lab, if you ever volunteer in a lab, you may be able to do the first few steps here, or you may be able to do just these steps here in the end. You won't be able to complete everything because anything in the lab just takes a lot of time. So science isn't super fast, sorry. So when you hear, oh, in 2 weeks, we're going to hear whether the Omicron variant, whether our vaccines still work against the Omicron variant without, taking human cases into account, it's 2 weeks because it takes 2 weeks to do the assays to determine this. It's not, so when you watch crime shows, it's often like, oh, I'll wait for the sequencing result. No, you don't. You don't sit next to a machine for 12 hours. So it's not that fast. So a lot of working in science requires patience and not getting too frustrated when things don't work, because 90% of the time you spend in the lab is time spent on 0 results. So in order to get an experiment to work, you often have to try multiple times. Okay, so this is just an overview about blotting techniques. So blotting is something I'll walk you through. Northern blotting, and then eventually probably through Western blotting. Blotting is a technique that we use to detect DNA, RNA, or antibodies. We have 3 different types: Northern blotting, Southern blotting, and Western blotting. You could be like, why are they called Northern and Southern and Western? Okay, so the guy who first did Southern blotting, who kind of invented the technique, was Edwin Southern, a British scientist, and the technique was named after him. So Southern blotting means you look for DNA by using a DNA probe. Now somebody else was like, oh, we can use the same technique for looking for RNA. Well, what name are we going to give that technique? Oh, it's Southern. Well, let's name it Northern blotting. So super creative as usual. So now we have Northern blotting. And then finally, when we look for proteins using antibodies, we have then Western blotting. There's no Eastern blotting. So anybody wants to, you can maybe look for metabolites using metabolites, and you could name that Eastern blotting if you want to. Nobody's done that so far. But it's an easy way to remember it. So DNA is the easiest one. So we'll use, that was Mr. Southern, and then we have Northern and Western blotting. Okay, so we will start with a Northern blot. If you remember, we have the patients. And if you remember, in human DNA, we have, so what we could do is just, isolate their DNA and sequence it. We would know what's happening. But really, you wouldn't know if the problem is alternative splicing, or if the problem is RNA processing, something like that. Some mutations that happen later on. So what you really want to look at is at the RNA that's later translated into the protein. So you can know what the actual problem in the patients is. So what you want to start with is with the RNA. The other thing why you want to start with RNA is the mRNA, the mature mRNA is much shorter. If you remember, in the DNA, you have all the introns. I think it was alpha tropomyosin was 200,000 bases long on the DNA level, and then the mRNA level was like what, 2,000 or something like that. So you want to work with a shorter sequence, which is why we'll start with the RNA. So in this case, you take what, a blood sample or whatever, and you isolate the RNA. You then separate the RNA by gel electrophoresis, transfer it to a membrane, and then probe the membrane. And I'll explain every step. I'll explain every single step of those. Again, if DNA was used, it would be a southern blot, but now we're using RNA, so it's a northern blot. Yep, question?
So it's RNA itself is mRNA, the mRNA.
Yes.
Even though they could splice it
We'll go through this. Yeah. Here your answer is coming in like 2 slides. Okay. So what tissue do we work with? Okay. So we'll start with isolating the RNA, and the answer, the question was, do we choose a specific tissue? We'll go and figure out which tissue to use by doing a northern blot to see where this RNA is even found, so that we know which tissue do we want to harvest a lot from in order to get the mRNA. Okay, if you want to separate mRNA, and this is something, there's DNA and RNA gel electrophoresis. It's a separation technique where we try to separate RNA by size. And it's fairly easy. If you remember, RNA is negatively charged, same as DNA, right? It has all those phosphates on there, very negatively charged. So we pour a gel like this one, and this gel lets smaller pieces go through faster and larger pieces take longer. It's just by size. If you have a small RNA or DNA piece, you go to the positive anode faster than if you are a larger piece. So what we do is we have a power supply, we have a little chamber, we have a positive anode and a cathode, which is negative. And we put our samples into these little wells here. So you have like little slots here that you can put samples in. So I take my RNA or my DNA and put it into these wells, and then I put electric current on, meaning the DNA or RNA, in this case, goes from these little wells all the way down here. If I let it run long enough, all the RNA, no matter what size it is, will go all the way through. Okay, because it goes to the positive charge. But if I just let it run for, and now this is something we've tried and tried and tried, if I let it run for 45 minutes, you'll have a nice separation. And what you get is small RNAs down here and large RNAs up here. So that's your first separation by size. You can then take, what do we do with that RNA? So RNA you can't really see. It's not something that's visible to the eye. And you can't really see DNA either. So we have chemicals that we can make it visible with, but if you can see it, but you still don't know what RNA species this is. So you can see, oh, there's a band of RNA, but do I know what RNA it is? It could be, myosin RNA, or it could be the cap protein RNA, could be any RNA. So in order to figure out where the RNA is that I like and that I want to see, I have to do what's a so-called northern blot. So here I have my gel that I just ran, and on top of it, I put a nitrocellulose membrane. So nitrocellulose is something that nucleic acids really like, and they bind to it without much ado. Okay, so you just need a reason for the RNA to go out of the gel to the nitrocellulose, and it will just stick to it, and it will not let go of it again. So now I can take my gel and transfer my RNA to the nitrocellulose membrane. And the way how we still do it, even though this seems so outdated, because we have machines for everything these days, we still do it by putting by using a buffer solution, an alkaline solution here, putting some paper towels down, putting the gel on top, putting the nitrocellulose on top, and then putting a bunch of paper towels on top. So what happens is just by the capillary effect, if you ever put a dry towel on a wet spot without even doing anything, the water will go up, right? That's the capillary effect. So just by that, the water will get drawn up. or the alkaline solution will get drawn up, and it will take the RNA with it, because the RNA has no reason to stay in the gel. It just goes wherever it's being drawn until it hits the nitrocellulose membrane. And when it hits nitrocellulose membrane, it sticks to it because it really has a high affinity to nitrocellulose. So now if I let this run, I let this go overnight, your RNA is now on this nice nitrocellulose paper here, okay, with tightly bound RNA. Okay, so now I have the RNA very nicely on my nitrocellulose membrane. There's several advantages to having it in there instead of the gel. So the gels are fragile, they break easily. They also don't bind the RNA tightly, right? So the RNA can just go in and out or move around a little bit, and it will slowly over time just diffuse into other areas. Once it's on the nitrocellulose membrane, it just stays there. We don't have to worry about it anymore. Okay, so now what does that help me? On the nitrocellulose membrane, I can now try to detect a specific RNA species, in this case, the FOX RNA, and try to detect it to see where it is on that, on my little nitrocellulose sheet. So if you remember, you had this with nucleic acid hybridization, I think you had with Dr. McLaughlin, that, DNA, you can denature it, make it, to have the double-stranded DNA and denature it. You can do the same with RNA. So RNA in your gel will form secondary structures. It will bind back to itself and form all kinds of nice double-stranded weird structures. You can denature it, meaning you can make it linear again, and then you can hybridize something else to it. So if you have just the regular DNA, this is just double-stranded DNA, but RNA kind of works the same, okay? So RNA in solution or even on a membrane is never just single-stranded, okay? Anything nucleic acids always looks for a partner. So it will bind back to itself. It might not be perfect binding, but it will still try to bind back to itself. So if you denature any double-stranded DNA or RNA and you cool it back down, it will re-anneal. This is a slow re-annealing process. It will find its perfect partner, everything's good. If you heat it up and denature it and then cool it really quickly, it will anneal much more imperfectly because it doesn't have the time to find its appropriate partner. For RNA, this is even worse. In the RNA, you don't have the perfect partner, right? You only have like partially complementary sequences. So the RNA will be even less re-annealed to itself. The advantage now is that you have some bases that are open and that you can bind something to that you put into the solution. And that's what we use when we do northern blotting. So we know that we're looking for the FOXP2 gene. We also know, because the human genome has been sequenced since the early 2000s, we know the sequence of the gene. What we now want to know, where on my membrane is my FOX gene? Okay, where's my FOX RNA? What size is it? Is it even there at all? We can now take a sequence of the known, like a section of the known FOX DNA sequence and make an oligo that's complementary to it. Okay, so let's say usually my oligonucleotide, which is like a short synthetic DNA strand, will be about 20 nucleotides long. So I can just tell a company, I'll send them the sequence I want, and they send it back in a little test tube. So you have this short DNA sequence that perfectly matches your RNA sequence. So when you're a student in the lab, you have to pay really careful attention to, remember which strand we transcribe and which ones we don't transcribe. You then have to make sure you have the complementary sequence to the mRNA. Okay, it's a little bit more difficult to design these oligos in the lab than it is when you just do it on paper. Okay, so you have this DNA strand that is reverse complement to your RNA sequence. We then label this, and we usually label it with radioactivity. You can also label it with a fluorescent label, or whatever else you can detect with a color label, something like that, just so that we can see it later, because just DNA or RNA we can't see. So then we have a radio-labeled oligo, which is now called probe. Okay, it's a probe because we're going to probe our membrane with it. Okay, so here's our membrane. This is the membrane that the nitrocellulose membrane that we had from the transfer of the gel that had all our RNA bound to it. We heat this up, then we cool it down really quickly in the presence of these radiolabeled oligos. So now, if in here, and it's in here, if in here you find the mRNA for the FOX gene, your oligo should be able to bind to it. Anywhere else, the oligo should not bind. It's short. So I said DNA and RNA really like being on the membrane. They do, but short nucleotides will not stick very long. So you incubate it with the probe, and then you wash away everything that's not really tightly bound to it. And what stays is the probe bound to your mRNA in the cell, in the membrane. Okay, how does this look like and why do we do this? If you remember, we have our patient and we want to isolate the mRNA so we can sequence it, so we can figure out what is the problem. Is there an amino acid that's wrong? Is there anything wrong with the splicing variant? What is the actual problem with the FOX gene? In order to isolate that, you first need to figure out where do I get enough material from, enough mRNA material from that I can actually isolate it. So we do these northern blots to see in which tissue do we have enough mRNA that we can isolate to then sequence and clone the gene. And this is what you see here, each band here. So this is an agarose gel that's been run. So up here are the higher up would be the larger protein, the larger RNAs, and lower down would be the smaller ones. So here on the side, you have the marker, which says kilobases. So this would be 9,500 bases long. This would be 7,500 bases long when you're about here, and down here, 4,400. So our band here that appears is at about, what, 6,000 bases. So now you can see in the heart, brain, placenta, and lung, we don't really have much of the RNA, but in the skeletal muscle, we have a lot. And in the pancreas, we have a lot. And we have, in the fetus, we would have a lot in the brain, lung, and liver. So now you know if I want to look what the actual problem is, I have to isolate RNA from any of these highly expressed tissues. So you would want to take a skeletal muscle. Now I can tell you this is a little bit of an unrealistic scenario, and it's a constructed scenario, because when you have a patient, you want to figure what's wrong with the FOX gene. I don't think you would do a biopsy on the brain for that. I don't think a patient with a speech impairment would necessarily want to have their brain biopsied. I don't think anybody would want that, just to see where it's expressed. But in general, this is what we do, and mainly we do this. We would use skin samples, blood samples, maybe saliva, which also has cells in it. We would use easy access material. We wouldn't, go and cut the brain open. All right, down here we have a control just to see if our northern blot works. In science, you always have to have a positive control and a negative control. So the positive control is the one that's supposed to work, that shows that you did your experiment correctly, that you didn't forget anything. And a negative control, which is something that's absolutely not supposed to work. So if you don't put any human sample in there, then there shouldn't be a band, right? If you still get a band, then you've done something wrong that showed up even though it's not supposed to show up. So you can't trust any of your other results. So always positive, negative control. Every single experiment in your life you'll ever do, don't forget the controls. Okay. As a side note, and that's not something that was done in this experiment, you can also do this in a large scale. You can do what's called a microarray, where every dot here represents a different gene, and you radiolabel the mRNA and then see if it binds to a microarray. So that's basically looking at the expression of, I don't know how many 100 dots there are, 500 dots or so, like 500 different genes. So these northern blots you can also do in a larger scale. Okay, you don't need to just do probe one gel at a time. I won't ask you how microarrays work, but it's a side note that everything can be scaled up. Okay, so now we know which tissue to take to isolate our mRNA. And now we'll go and try to get it to be a DNA, because as I mentioned before, you can't really work with mRNA because it's always so unstable or unstable. And you also can't make protein from an mRNA. Well, you can, but it's not, you can't make a ton of it. Okay, if you just start with a little bit of mRNA, you can only make a certain amount of protein. Whereas when you start with DNA and have your bacteria work for you, you can make a ton of it. So how do we get RNA to be a DNA? We make a cDNA. So this is a complementary DNA. A cDNA is not the same as genomic DNA. A cDNA is the copy of the mRNA, so it doesn't have any of the introns. Okay, so that's really important to remember, that when you work with when you make a complementary DNA to an mRNA, it's not identical to genomic DNA. So you will have databases with genomic DNA sequences and some databases with cDNA sequences. They're not, like, the information in cDNA sequences is different. This is your readily spliced done transcript, mRNA transcript, versus this is your whole genome. So we start with a total total mRNA prep, and then we synthesize the cDNA using a reverse transcriptase and a primer. Then we'll degrade the RNA, and then we'll synthesize the second strand. I'll walk you through that. Now, this is, remember how I said that information always flows from DNA to RNA to protein, and we can't go back from RNA to DNA? Humans cannot, okay? Humans cannot go from RNA and make it into a DNA, but some viruses can. So when you have an RNA virus it encodes a specific protein called reverse transcriptase. And this protein can use RNA as a template and make it into DNA. Okay, so this is here, it's called reverse transcriptase. So there is a way of going from RNA to DNA, but as I said, humans cannot. So again, an mRNA vaccine cannot be reverse transcribed into DNA and integrated into the genome in humans because we don't have a reverse transcriptase. Okay, so how does this work? You have your mRNA here, and you can remember you have the 5' end, and then in the end you have a poly A tail. You also know that mRNA goes 5' to 3'. Now, if I want to reverse transcribe and make this into a DNA, the same rule that applies to any nucleic acid polymerization applies here too, that you have to go 5' to 3'. So anything that I want to make here has to start from this end here and be transcribed, reverse transcribed into this direction. Now, the cool thing about mRNA is all of it has a poly A tail. So if I want to just take the whole mRNA out of your cells and reverse transcribe it, all I need to do is get a poly T primer. So the reverse transcriptase is similar to the DNA polymerase. It needs a primer to get started. It can't just start without having a primer in there. So here's your DNA primer that I can just buy and add to the mixture. And now the reverse transcriptase will make a cDNA strand all the way to here. So now you have one single strand here. Now this is unspecific because you have a poly-DT primer. I'm not just amplifying my FOX gene. I'm amplifying all mRNA, but only once. Okay, so I'm going once and make one copy of every mRNA in here. What I'm also doing, if there is, let's say, 1,000 copies of the FOX gene in there, I'm making 1,000 copies into cDNA. So that's why I wanted to isolate a tissue that has a lot of the mRNA, so they have a lot of starting material for my reverse transcription. Okay, now we have the problem that if we want to amplify, or if we want to put the secondary strand of all the mRNAs, we get an mRNA library or a cDNA library. We don't have a primer for this side, right? So we now have a DNA strand here with a T tail in the end, but if I want to replace the red strand and don't know the sequence here, or just want to get all of it, how do I start? We need, for now, a DNA polymerase, we need a primer again. The way how we get this primer is by using RNase H, which is an RNase, and we add DNA polymerase and DNA ligase, and then we only partially degrade the RNA. So in this case, we keep all these red sections of RNA here, and this is a random process. So we partially degrade the RNA, meaning we take out sections of the RNA, but some RNA sections are still bound to the newly made cDNA piece. Does that make sense? I know it's not the easiest, but okay. So now we have primers, and then we can amplify the sections in between. That's kind of the same how you had in DNA replication when you have an RNA primer to start with. Okay, you then have, in the end, you use DNA ligase, which will ligate the blue ends together. Okay, so in the end, you have a double-stranded cDNA with just a little section missing up here, because this section is the one that we don't have a primer for. Right here, this section's missing. Okay, so in the end, what you have after all this, after all these steps, you have a mixture of all the cDNAs from an mRNA tissue, from the mRNA from a specific tissue. So again, we have no, so far our experiment is completely unspecific. So you can now amplify from that. You can try to pull out the Fox mRNA, the FOXP2 mRNA, or you can try to pull out any other mRNA that you want to work with. Yep, question. So if you would stick with this, where you have the cDNA and mRNA, you couldn't replicate it, right? So in order to get a DNA that you can work with in the long run, you need a double-stranded DNA so you can replicate it later. Yes, question. Yes, so the partially degraded mRNA is used as a primer to do the 5' to 3' elongation with the DNA. Okay, so your little RNA sections have a primer. Yes, question?
And those so-called primers are eventually degraded by RNA.
Yes. Yep. So another way on how you can do this and how we often do it is if you know the sequence you want to work with and you don't care about all the other mRNAs, you just add a little DNA primer here. So you degrade the entire mRNA and just add a little DNA primer here to this end, right? So instead of having this red section here, you add a no, and I know the sequence, I can just order a short 20 nucleotide primer, and then the DNA polymerase will amplify this specific gene. This wouldn't be a cDNA library. So this now is called a cDNA library because it has all the different cDNAs from that tissue. If you go and do one specific DNA only, you get one clone, which, you know, in this case would be sufficient, but in other cases, you do want to see all the genes that are expressed. Okay, so now we're here, we synthesize the cDNA, and the next thing we want to do is we have a cDNA, or we have all the cDNAs, but really to work more with it, we need a lot more. So just having 1,000 copies of that cDNA is unfortunately not good enough for what we want to do. So we need a lot to be able to go through all these other steps that we want to do here. So you just need a lot of starting material so something works out in the end. Okay, so the method how you get a lot of DNA from just a little bit of DNA is called PCR, polymerase chain reaction. So PCR was a huge breakthrough in the 1980s. I know that's long ago, and even for me, that's long ago. I wasn't working in a lab in the 80s yet, but for the people who did, this was a super exciting breakthrough because all of a sudden you could make from a very little bit of DNA, you could make a lot. So the other things that PCR did, not just help with lab work, but for the first time we could do forensics, where you have a tiny bit of a blood sample and you're supposed to do a fingerprint of something. So you need to be able to make it more because these little bits we can't visualize. Paternity tests also for the first time were determined, or were gaining tractions in the 1980s. Before that, all we could do is blood type. So you couldn't really tell anything from the DNA because we just didn't have enough. And yes, I mean, and you could just take a whole human brain to get enough mRNA to do something, but usually patients don't like their brain taken out. So it's good to have PCR. Okay, this is how PCR works. So PCR, now we want to just amplify our Fox gene, right? That's the one we care about. We don't care about anything other than that. What you need to know for PCR is the sequence you want to amplify. So you cannot amplify things that you don't know the sequence of, which is a big drawback, but that's just how it is. So this is how PCR works. This is your target sequence in your DNA here. And you have the 5' to 3' one strand, and the other strand also 5' to 3'. Regular DNA that we just made that DNA, okay, from the mRNA. So now you order 2 primers. Primers are, as I said, those 20, I don't know, between 15 and 30 nucleotides long, depending on what you got, that are exactly reverse complement to the sequence that you want, okay, the sequence that you know. So if I know my gene starts with ATG CCD, or well, it's not D, ATG CCG, then that's your primer, okay? So you want the exact matching sequence to that. So you order a primer that's reverse complement to your 1 DNA strand here, and you order another primer that's reverse complement to your other strand here. So you have to separate the strands by denaturing the DNA at 95 degrees, and then you cool it down to about 50 to 60 degrees. At about 50 to 60 degrees, your primer will anneal at this little sequence, but your whole DNA strand will not quite anneal yet. Okay, so you take advantage of this shorter sequence that it anneals first to your DNA. The other thing you do is just put a ton of that primer in. So the likelihood of your primer annealing rather than your 2 strands back annealing is just much higher. Okay, and then you needed a DNA polymerase that it can extend your primer and make it a full second strand. So in this case, we use a thermostable DNA polymerase. The one that we use a lot is called Taq polymerase. So that's from Thermos aquaticus, which grows at 72 degrees. I can't talk, sorry. So Thermos aquaticus polymerase likes high temperatures. It's an organism that grows at high temperatures. The other thing that polymerase can do, it can tolerate temperatures up to 95. So you keep it in this mixture where it can work at 72, but it also survives at 95. So it doesn't really care about the high temperatures. You couldn't do this with a human polymerase. Human polymerases would just denature and not work anymore by anything over probably 45 degrees. Okay, so you need your primers, you need nucleotides, the NTPs, obviously, to do your replication or your primer extension. And then you end up with these 2 products. And you can see this product starts here because that's where your primer started and goes to the very end of the template that you have. And the other product starts here and goes to the very end of this product. Now you do the same again. You go to 95, you denature the DNA, meaning you take the parental DNAs apart, you anneal the primers, and you extend the primers. And this is how the second cycle looks like. You now have, some of the templates are already shorter because you have the shorter template, and some are still longer. So now in the second, after the first cycle, you come up with those 4 different DNA templates. The longer parent here, the longer parent here, and then 2 shorter versions here. Now you amplify these with binding of your primers, you end up with these really short regions that are the ones that you're interested in. If you repeat these cycles over and over and over again, you start getting more and more of your short sequences. So we do, in cycle 3 is the first time that you get your target sequence amplified in the right length. And now these different ones are going to be your targets for the next round. Now, over time, the shorter ones are going to take over. So you can, if you draw this out for cycles 4, 5, and 6, you will notice that you have more and more shorter products that are the correct lengths that you want. And in the end, I can tell you, when you do a PCR, if it works, doesn't always work, but if it works, you only see a band of your shorter product. You don't even see the higher up ones or the bigger ones, because you do this 35 times, you never get more full-length product than what you put in, right? Because you can never go back to that, but you get more and more, exponentially more of the shorter product. Okay, and then what we do is we put this on a gel, we look for the band of the right size, and then we cut it out. And then we have a little piece of gel with the exact DNA that we want to have. Okay, so now you've successfully isolated the DNA of the FOXP2 gene. Okay, this is just another side note. There's many different applications of PCR. You can quantify how much of your target mRNA is in there. So I can look how much mRNA of certain gene is in the brain, similar to Northern blot, but you can do it with PCR as well. You can do DNA fingerprinting. You can compare samples from different people. So again, you're going into forensics and you're going into paternity tests. And you can also, by making a mismatched primer, a mismatched oligonucleotide, you can introduce mutations. So you don't need to memorize this, but it's nice to know that PCR is really what revolutionized a lot of our molecular biology techniques. Yes, question.
So I'm not sure if I missed this before, but what's the significance of having cDNA rather than gDNA? I know like the introns aren't there, but why is that important?
Okay, so if I want to, the question was, what's the significance of using cDNA rather than genomic DNA? If you use genomic DNA, the one thing that you have this huge section of DNA that's harder to work with. On the other side, if I want to express my protein in E. coli, and we'll do that at the end of the whole section here, E. coli doesn't have splicing. So if you want to make your protein in a bacterium or in a yeast that uses a different splicing mechanism, you can't put the pre-mRNA in there because the pre-mRNA would have all the introns. And what's E. coli going to do with an intron? It doesn't know anything. So it'll just not make the proper protein. So if you want to make the protein in a different organism, then you have to get the cDNA. Okay? All right.
As a follow-up, okay?
Yeah.
In DNA fingerprinting, I know that they use things like ENTR, stuff like that.
Yeah.
Aren't those also found in
So DNA fingerprinting, you could use genomic DNA.
Oh.
So this is a applications of PCR. You can use them for genomic DNA, cDNA, whatever. So this specific experiment that we're doing, we're using cDNA, but often, yeah, for paternity tests or for forensics, we use genomic DNA. Okay, so now we have made cDNA and you've learned about PCR. What we'll do now is we'll take our DNA section here and put it into a so-called plasmid. So E. coli and other bacteria have their chromosomal DNA, and then they often have so-called plasmids, which are short circular DNA strands that kind of, they replicate independently from the chromosome in the E. coli. We'll put our gene in there, and then we'll force E. coli to make the protein for us. So these are plasmids. So this is a bacterial cell, and this is E. coli, very nice drawing. It has its own chromosome, and then it has these optional plasmids. So not all E. coli in your gut will have plasmids. Sometimes they will have several different ones, and sometimes they just have a few hundred of a single species of plasmid. So they're short, circular little DNA sequences that kind of float around in the cytoplasm. And we can make use of these to introduce foreign genes into E. coli. So molecular biology has hijacked these plasmids for our own purposes because messing with the E. coli chromosome is complicated, and using plasmids is incredibly easy. Okay, so this is a plasmid feature. So in order for these plasmids to be able to survive in E. coli, you need an origin of replication, which tells E. coli to make more of it, because every time E. coli divides, it would need several to go into each cell, right? So we need an origin of replication so that the DNA gets replicated and that the plasmid gets maintained in your bacterium. In order for us to force E. coli to do our job and make the protein, we also need to force E. coli to keep the plasmid. So if I were just to try to have E. coli make a protein for me, it would just kick the plasmid out, because why would it make all that protein if by kicking the plasmid out, it doesn't have to do it? So what I put on there, or what people have put on there, is a so-called selectable marker. And in this case, this is an ampicillin resistance. Ampicillin is an antibiotic that will kill E. coli if it doesn't have a resistance. If it has a resistance gene, so this resistance gene could be something that cleaves the antibiotic. something that pumps out the antibiotic. There's several ways of how resistance can work. If it has a resistance gene, it will be able to survive. So this one has an ampicillin resistance. And what this gene does is if you put antibiotic ampicillin in the media, the ampicillin goes into the cell, the resistance protein is able to cleave the ampicillin, making it not work anymore. So if E. coli has this AMP resistance, it can survive in media with ampicillin. If it doesn't have it, it dies. So now it's forced to keep the plasmid in the cell in order to survive. So that's how it's called a selectable marker, because I just kill all the ones off that don't have it. Okay, so it's a fairly easy way on how you can manipulate an organism to do what you want it to do. Another thing that the plasmids that we use have is a multiple cloning site, and this is basically just a region that has a lot of restriction sites in it, and I'll explain to you what restriction sites are. Restriction sites are short DNA sequences, and they're usually palindromic, meaning they are GAA, TTC, and then if you read it from the other side, it's the same, GAA, TTC. So same here, GAT, ATC, and then you read it here, GAT, ATC. That's palindromic, both sequence on both strands. So they're short sequences that are recognized by so-called restriction endonucleases. If we have those in the plasmid and we have a restriction endonuclease, I can cut the plasmid open. Okay, so it allows me to manipulate the plasmid at a specific site by having an enzyme that can just cut it at a, and it cuts it at a site that I wanted to cut it, not in like the ampicillin resistance or anywhere like that. Okay, so I'll walk through a restriction endonucleases and then we can stop for the day. What restriction endonucleases do is they make double strand breaks in the DNA. So this is your plasmid would go around here, and you have this little sequence, and EcoR1 is an example for a restriction endonuclease. It would cut here and it would cut here, and what that generates is a DNA that looks like this. So you have the one strand ends in a G and then has a 5' end overhang here, and the other one starts here at the A and has a 5' end overhang here. Here, this is your 5' end overhang. Depending on how they cut, you can get different cut patterns. So if it cuts here and here, you get a so-called 5' end overhang, because this is still double-stranded here, and then your 5' end extends. You could, if it would cut here after the T's, then you would have a 3' end overhang. So that just depends on the restricted endonuclease. And the sequence is specific for the enzyme that you're using. If it cuts straight through, that's called a blunt end. Whenever it's straight through, it's blunt. When it's not straight through, it's a sticky end. And sticky ends can be 5' overhang or 3' overhang. Okay, so now you have those different restriction endonucleases. Some make blunt ends, some make sticky ends, and then you want to put it back together. Now, the important thing in putting back together is that you can either put two blunt ends back together, then the sequence doesn't matter, or you have to have what's called compatible ends. So if you have an overhang here, this TTAA will only go back together if it gets an AATT, so that the A here can base pair with a T, and this A here can base pair with this T. So you have, for sticky ends, they have to be compatible. If by chance you don't have compatible ends, you can either use a polymerase and fill it up. So I can just use a regular polymerase that extends from 5' to 3', give it some nucleotides, and then it'll end these nucleotides to make a blunt end. But you can only do that when you have a 5' end overhang, because nucleases, DNA polymerase can only go in one direction. Or you can cut away the extra ends with an exonuclease and also get to a blunt end. So either compatible ends that stick together or blunt ends. Those are your only ways of putting the DNA back together. And with that, I think we'll do the cloning and how to get it into the plasma next time, which is Monday. So I wish you guys a happy weekend.